Welcome to Under the Microscope. This series is brought to you by the Real Scientists Nano team. Our goal is to provide a platform where scientists can communicate their work and interact with the public. With that in mind, every week we introduce you to a scientist working in the field of materials and nanoscience. Hello everyone. Today we have with us James Armstrong, who is a research fellow at Imperial College London. Hi James, how are you doing?
Hi, I'm very good, thank you.
Okay, wonderful. Let's understand your research a little bit. So could you explain your current research to us in super simple words, please?
Sure. So I'm a tissue engineer. So this basically means that I use different combinations of cells and materials and chemicals to grow artificial tissues in the laboratory. So these can then potentially be used to replace diseased or damaged tissues, or they can be used to study different disease processes or to screen drugs. A big problem that I'm trying to address here is how to recreate the natural structure of these tissues, because in biology, structure is equal to function. And if we're going to create functional tissue constructs, we need to be able to create natural structure. So to do this, I've been developing a new technique. It's a bit left field, and it uses sound waves to remotely organize cells. And it does this to present them into their natural configuration. And then I've been using this in particular to create structures of muscle tissue with aligned cells and fibers. Again, by presenting the cells in their natural structure, we can create more realistic, more functional muscle tissue.
Okay, that sounds interesting. You mentioned you use sound waves to align the cells to form the tissue?
That's correct, yes. So we actually use ultrasound. So we use a particular kind of ultrasound called an ultrasound standing wave. So we create regions of high pressure and regions of low pressure, and these are fixed in space. And the cells essentially don't like to sit at the regions of high pressure and low pressure, so they move to the regions of unaltered pressure, let's say. And then we can create these different pressure fields to basically position the cells where we want to. And we can do this in 2D and we can do this in 3D.
That is so cool. I'm really curious how it works, especially in 3D with the sound waves, how you can control the pressure, because it must be challenging, right?
So 3D is a big challenge, actually. People are starting to explore this, but we basically use sources of sound. So it's a little bit like having speakers. And to create the standing waves, you just need speakers which are facing each other. So to do this in 3D, we sort of need a speaker at the bottom and the speaker at the top. And this creates sort of a stack of pressure waves. And then what you can essentially get here is the cells levitating into those different pressure regions.
Okay, that sounds super interesting. So let's get to what can the followers expect in the week that you will curate the RealSci_Nano Twitter account?
Sure. So I guess I'll just start off on Sunday just talking a little bit about myself, who I am, the sort of the general bits of research that I'm interested in. And then Monday through to Friday, I think I'm going to talk about in a bit more depth about the different areas of research which I'm passionate about. Monday, I'm going to start off with some of the work I was doing in my PhD and some of the techniques surrounding painting cells for tissue engineering. On Tuesday, I'm going to move on to some work where we can use magnets and magnetic fields and use these to guide tissue engineering processes. And then Wednesday and Thursday will be my main focus, as I've already discussed, in terms of using sound waves, using these to build tissues. But then also on Thursday, I'll talk about how we can use these to stimulate, activate enzymes and trigger the assembly of materials. Friday, I'll talk a little bit about 3D printing of tissues and other similar biofabrication techniques, how we can use very precise manufacturing processes to produce living tissues.
Mm-hmm.
And then on Saturday, I think I'm just going to take a bit of a deviation, and I'm going to talk about something which I'm passionate about at the moment, which is the availability now of online seminars in 2020, which I think is really important given all the cancelled conferences that's been happening due to the present circumstances. I'm curating and chairing a conference which I will talk a little bit about, but I'm also going to highlight some fantastic opportunities for people out there who are doing science and want to see some of the great science going on and maybe have an opportunity to present their work and network with people in the absence of any conferences.
Okay, that sounds wonderful. It's an action-packed week,
Yeah.
and we are looking forward to the week you will curate the account. And thank you very much for speaking with us.
Thank you.
Thank you for listening. To know more about us, please visit our website, realscientistsnano.org, and follow us on Twitter at realsci_nano.